thing, that is, now we are starting to go on space missions very frequently. So as you can see on my screen, NASA has already planned the Artemis mission, that is the next human landing mission on Moon after the Apollo missions. But unlike the Apollo missions, these Artemis missions are not going to be a 2-day affair. These astronauts are going to stay on the surface of Moon for months, at least 6 months. And quite naturally, the effects of this extreme journey and this settlement on the surface of Moon is going to have some effects on the body of the astronauts. So this is something which I am really interested in looking into and understanding more about the mechanisms that govern these changes. Also, beyond Moon, we are definitely going to Mars sometime, maybe in the 2030s or the 2040s, because as you all know, this person, Elon Musk, would be very disappointed if we can't do that, just like he said in his quote. So with Starship being ready at a very rapid speed, it looks very likely that human beings would be traveling to Mars. Also, another aspect of space travel which has seen a huge boost in this year itself is space tourism. With Blue Origin and Virgin Galactic coming into the picture of space travel and doing some successful space tourist missions, you can understand that now more and more people are going to be involved in spaceflight travels, be it an astronaut or be it a civilian. So it is quite the high time to look into the problems that is faced by the astronauts who go to space. Some of these problems are already known to us since the days of the Apollo missions, that is almost 50 years ago, like bone and muscle density loss. That's the reason why the astronauts aboard the International Space Station have to indulge in rigorous exercise daily to maintain the bone and muscle density. Otherwise, they lose it at a much rapid speed. Definitely, there's physical strain, fatigue, but there are certain molecular changes as well, and those molecular consequences are not visible to the naked eye. You cannot see the phenotypic changes at first, but those changes do occur, like what Dilara was saying about the changes in the gut microbiome. And factors of spaceflight like radiation and microgravity play a big role in changing this gut microbiome composition. Then there's risks for cancer development because of certain signaling pathways being altered. There can be reproductive system issues, neurodegenerative diseases, immune response changes, metabolic changes, and who knows what else. There are so many more unexplored issues which you and me can look into in much more detail. So, one of the major landmarks in the study of what happens to the human body at a molecular level was studied in the NASA's twin study. This was a boon to humanity because we got an unparalleled opportunity to study the effects of spaceflight, the various factors like, as I said, ionizing radiation, microgravity, atmospheric temperature, atmospheric pressure composition difference, and many more, on two identical twins. These served as the best samples that you can get because the molecules, the genes, everything is same. So the Kelly brothers, Scott Kelly and Mark Kelly, were involved in this experiment, and Scott Kelly went to space and spent over 300 days on the International Space Station, whereas Mark Kelly stayed on Earth serving as the ground sample subject. After Scott Kelly returned to Earth, as Sonalika mentioned in the introductory part, there were 9 different groups who were assigned different omics studies. There were groups which focused on the genomics, the transcriptomics, proteomics, metagenomics, epigenomics, and there was one final group whose duty was to integrate all the omics and analyze the cumulative effect on the genome of Scott Kelly, and they were actually looking into multi-omics. So at Pine Biotech, we focus on all these omics studies, including multi-omics, and how we do it, I will be showing you in a while. But first, let's talk about what were the results. What did we observe from the twin study? So to begin with, let's talk about genomics. So in Scott Kelly, there was a noticeable increase in telomere length, which was kind of not expected because as we know that telomeres should be shortened and aging is a problem of spaceflight, but quite interestingly, during his mission, it increased, but once he returned back on Earth, it shortened drastically. So, this sort of observation needs more research and understanding. Similarly, talking about transcriptomics, there were various gene pathways that were altered, various novel genes that were identified to be affected by spaceflight, and scientists are still looking into what effects does those alterations have on the astronaut's body. So there are several other studies or several other omics results which we will talk in pretty much detail in the program, where we will talk about the NASA's twin study as a case study. We will take every omic level and discuss in detail the results, the findings, and the implications, and what can we do with these results in the future? How can we get involved? But this is the time when we should mention what made this sort of research possible. So we are here dealing with NGS data or next generation sequencing data, and that's how we can actually look into the genome and the transcriptome. But this genome sequencing was made possible by Oxford Nanopore Technologies MinION, which is a very small sequencer, the size of a pen drive. It can fit into your pocket, and this was taken aboard the International Space Station, and the astronauts' blood was drawn and sequenced right there on the International Space Station. And this technology has opened the door for a new era of space omics, which we are dealing with in this program and also in the upcoming 1 month program. Now, I would give you some example of the different omics studies based on space flights. One of them is the transcriptomic analysis of mice, and these mice were subjected to simulations of deep space exploration. For example, if you would like to go on Mars, then simulating that kind of a travel for a longer period of time and to see what are the effects on the transcriptome, on the gene expression of mice, was the focus of this research group. And they found various immune system pathways and microRNAs to be differentially expressed. Now, what is differential expression? It is the change in the amount of RNA that translates into protein under two different groups. So this sort of a change usually has a lot of effects, and those effects are very fatal sometimes, like they can cause cancer or they can cause other diseases which are pretty dangerous. But it's not all sad and sorrow after all. And this particular research deals with cancer cell lines which are cultured on Earth and then taken to space and kept in the International Space Station. Interestingly, it has been noticed that when these cancer cell lines are kept in microgravity, they lose their potential of signaling. They stop signaling between one another because there's no gravity to keep them together, most probably, and they form this sort of a spheroid, which is called a multicellular spheroid. They start rotating and they lose their capacity of signaling. And this loss of signaling potential stops metastasis. Metastasis is that stage of cancer when the cancer starts to spread from one particular part of your body. For example, if you have lung cancer and metastasis kicks in, it starts spreading all throughout your body. But it has been noticed that microgravity acts as a boon or a gift, and these cancer cells stop proliferating. And maybe this is the beginning of something wonderful in the domain of cancer medicine. And that's why you must have come across the term space medicine a lot. So space medicine is not only something which deals with taking care of astronauts and protecting them from harm, but also to uh, understand and analyze various treatment technologies and methodologies about the International Space Station, use this unique environment of space, and how can that be utilized and brought back to Earth for the better good of each and every human beings on Earth. So this is the reason why we are talking a lot about space omics. Space omics is not only a niche subject, but it has the potential to be spread all over the world and to benefit each and every human being. And if this isn't interesting to you, then I don't know what is. Now, I know many of you who are sitting today in the audience are interested in getting involved in astrobiology, to get involved with some great organization, to do some internships, to get a start in the career, right? So here's a list of certain opportunities that I'm going to talk about, so, which would definitely benefit you. The first one is the astrobiology school, which is known as RED, Recontant Exobiologie pour Doctorants in French, or the Exobiology School for doctorates, for PhDs. So this is an opportunity for PhDs as well as some master student as well, who can participate in the French Exobiology Society's course and get involved with great ESA scientists, that's European Space Agency scientists. I had the opportunity to interact with them one on one and get some vital insights. That's an amazing opportunity. Then you also have Blue Marble Space Institute of Science, which provides young scientist program as well as visiting scholar program opportunity. The young scientist program is for bachelors, and the visiting scholars are for masters. And those who are beyond that, maybe you are pursuing a PhD or postdoc, or you are a faculty or a scientist, you can still join BMSIS as an associate research scientist. I will talk about the criteria and the requirements to get into it in a while. So you can also get involved in some analog missions such as ESEP, Earth Science Exploration Program. It was done in Ladakh, and it is going to be done over and over again. So you would definitely get the opportunity again. This is done mainly by Amity University, Mumbai, the Amity Center of Excellence in Astrobiology. Then there's a Lunaris analog research station. It's a moon analog where you can spend a lot of time, do some experiments, gain vital insights, some results, and the environment, the feel is as if you are on moon. You can also get involved in the European Astrobiology Institute, LC Japan, that's Earth Life Science Institute. But more importantly, you can also showcase your projects, showcase whatever you have done, and interact with some great scientists in conferences. So there are a number of conferences which are held every year dedicated to astrobiology. For example, AbGrad-E, this is a conference for astrobiology graduates all over the world. The students can participate, whether you are an undergraduate student or a graduate student, whether you're doing your PhD, you can participate here. There are some great keynote lectures by some great astrobiologists, so you can listen to them, get vital insights, interact with them. And there's AppSciCon, which is hosted by, in collaboration with NASA, and it's a NASA funded conference. So this is going to be held in 2022 as well. So definitely check out AppSciCon and AppGrad-E and all of these institutes and their opportunities. Now let me tell you the major requirement to be a participant in all of these events. So all of these events, as you might have guessed, requires you to be involved in some sort of a project, an astrobiology or a space omics project, so that you are excited, you are interested, but you are going to attend these events to do something more, right? You are not visiting a TV show or maybe some YouTube video, right? You are going to a conference or an event. So you are expected to do something greater, a little bit more than what you have done. So doing a project, at least starting a project of your own, is the way to go, is the first step that you should take. And here we are, Pine Biotech is gonna help you to get started with your own astrobiology or space omics project. And how to do that and what is the procedure, I'll talk in just a while. But before we move on, let me give you an overview of the career path to become an astrobiologist suggested by NASA. So this is the most authentic data that you can get, right? You don't have to trust my words. You can believe what NASA has to say. But before I talk about this, it would be great if you can head over to the chat section and write down the level of education that you are currently pursuing, whether you are a bachelor student or a master student or a PhD student, or you are a postdoc, or you are a scientist. And it would be great if you can also write down what's the subject that you're studying, so that I can get an understanding of what kind of audience I have. And I would show you that how whatever you are studying, whatever field you belong to, whichever subject is you are majoring in right now, you still have a great chance to become an astrobiologist, and the career path is pretty straightforward. So I can already see some great responses. I can see,
Sorry
yeah,
to interrupt in between.
yeah, sure,
Here to your question as well, I had a poll with me which I was supposed to launch. So it would be actually very great if I just launch a poll and
yeah,
everyone could just vote and tell us to which different fields they belong to and also what are their research interests. So this way we will have saved the different sorts of fields and research interests of our participants. So my first question here is,
absolutely.
what is your level of education? Are you an undergraduate, a graduate, a postgraduate, or pursuing PhD, or done with PhD, or a postdoc? And also, what are your research interests? Omics, space omics, infectious diseases, or data science, or precision medicine, or bioinformatics using R and Python, or machine learning? And in the chat box, you can feel free to tell about which particular field or degree you can say you belong to. Over to you, Shubhraji. Thank you. I'll be just closing this poll in 30 more seconds.
Awesome, great. Thank you, Sonalika. This is a great way to learn your responses and understand you, to know you well. So it would be great if you can go down to the poll and register your answers. And later on, I would definitely listen more to your interests, interact with you, answer your questions at the end of the webinar, and maybe then we can discuss further. So I could see a lot of people who are currently pursuing undergraduate. They are from biotech, they are from pharma, they are from microbiology, some are masters, some are PhDs and postdocs. So here is your career path as suggested by NASA. So you are earning a degree right now, right? So you're in this particular domain. And NASA says that you can earn a degree in any field. It can be physics, biology, oceanography, geology, astronomy. Definitely, biology also includes medical science and other subdomains. So you are earning a degree. At the same time, you're also building a community. Talking about the community of astrobiologists, since astrobiology or space biology is still a very new term, the community size is very small, but it's great to see that the number is increasing. Today we have 77 people who are enthusiastic about astrobiology here. There might be many more attending the YouTube live sessions. many more people who might not be able have been able to join because of the time zone difference, but they would definitely get a recording of the video. And so this community is growing, and this is very important in your journey, earning your degree and being a part of a community, so that you can interact with people, develop projects, work on them, and understand how uh, an astrobiologist works. So that's what NASA says. You do your graduate work, you join various societies, clubs, or organizations, just like you are today here at Pine Biotech, being one of those organizations. And you can earn your doctorate, that's a PhD degree, look for funding opportunities, and there you are. You are almost there. You are now having a fruitful career in astrobiology. I will give you a fun statistics. Today I went over to LinkedIn. I guess many of you use LinkedIn. If you don't, please make an account on LinkedIn and feel free to connect with all of us, and we can chat and discuss more about space omics there. So I headed over to LinkedIn, and in the job section, I filtered the search with astrobiology as a term, and I came across around 20 jobs available in the domain of astrobiology in the last 24 hours, I guess, which has been posted. So you can see there's a growing number of opportunities in the domain of astrobiology, and the people to grab them are few. So this is your great opportunity to make yourself available, make yourself aware, and kind of get your hands dirty in this field, so that you would be selected above others and you can begin your career in the domain of astrobiology. All right, enough said about career opportunities, but the main question which comes up is how to start working with the data that is generated from the spaceflight missions, right? What is the mechanism or the methodology to work with this data? Well, that is something which this program is going to talk about, and we are going to hold your hand, take you step by step, and make you understand how to work with this data. All of the projects, all of the research that we do at Pine Biotech is done on the server of Pine Biotech, which is called the Server T Bio Info platform. I'm going to give you a quick overview of what this platform looks like and how you can work on this platform, right? So, um, if you can see my screen, then I think now you would be able to see my screen with the server tbioinfo platform. And I would also, all right, thank you, Navya. So there you go, you have the link in the chat section. So head over to the link, click on the link, go to this particular page. You might get a prompt to sign up. Just sign up using your email ID or Google email ID, and you will get the uh, credentials to use this pipeline in some time. And now I would show you what this server platform is all about. So, the data that is generated from any experiment, be it a space related experiment or a much focused biological experiment, it's huge in its volume. There's big numbers of gigabytes involved. So it is not possible to analyze them on a personal computer. Also, if you would like to use your coding skills, it would take hundreds of lines of codes, which is not always a sweet spot for biologists, right? So that's where Pine Biotech's Server T Bioinfo platform makes things easier for you. If you would look on my screen, you would see that there's various categories in which the server is divided. You would find the NGS data, mass spectroscopy, that's related to proteomics, structural biology, data integration and modeling, which mainly deals with machine learning algorithms, virology, cancer biology, and a lot. Under the NGS data, you would see there's transcriptomics, genomics, epigenetics, DNA, RNA, and you see a lot of names, right? So I'm gonna go ahead and show you how these platform works. So if you look at transcriptomics, the first option is an RNA-seq, and there's a drop down button. If I click on here, you would get some demo pipelines. So let me click on one of the demo pipeline just to give you an overview. So here's what the platform looks like, and all these names that you can see here are basically the algorithms which people use to analyze the data sets. But to actually use this algorithms in your computer using code would be a very tedious task. But Pine Biotech has made it easier for you. How much easy? Well, as easy as just a point and click. So it would just take you a click to use these high profile algorithms to analyze your data. And that's not the end. As you can see, like magic, there are certain algorithm names which are highlighted in yellow. So this platform is AI enabled, and it gives you a hint as to what should be your next algorithm in the process. So in a typical RNA-seq pipeline, you have the next generation sequencing data which is generated by the sequencer. You then do some pre-processing, that is cutting off the unwanted reads. You can see here it shows you how you just chop it off which are unnecessary. You also do some cleaning, PCR cleaning, which are unwanted. Then you go on to the next part, which is alignment on the genome. Then you form some alternative splicing, isoform construction. To those people for whom these terms are pretty unfamiliar and new, I have a secret tool for you and how you can actually get hold of these terms. And not only that, how can you also learn to use this platform? I would tell you about that in just a second. So once you click on end and you make like connect all the required algorithms and a beautiful visualization here, by the way, you get that output. Since it's a demo pipeline, it generated the output right away. But if it would be a real pipeline which you would use for your project, it would take a lot of time because it's very server loaded thing. All right, so these are the various results that are generated from the pipeline. What each of these means is might be something which is new to you, but I would give you again in a while a trick how to understand that. So maybe I downloaded this particular file because this is what I'm interested about, and here you I have downloaded it. You can see, I guess. Just a second, I will share my screen well so that you can see it. All right, so here you can see that the data which I showed you has been downloaded here, and then I went on to load this particular data in an Excel sheet here. So in this Excel sheet, you can see that table says that there's group 1 of certain samples and there's group 2, and what you can do is you can compare the various genes and their expressions. Here are the gene IDs, as you might say. So this is the entire work or the research that people out there do, but now, as I promised, I would show you how you can learn about them. So if you go to the Learn OmicsLogic platform, I'm sure you would get the link in the chat section. You would get various options here. If you click on courses, you would see there's various courses, just like Sonalika mentioned in the introduction part, right? So here's an introduction to bioinformatics. There's bioinformatics using Python and various other courses like genomics and transcriptomics. If you go down here, you can see various projects, guided projects, which you can do just to get warmed up. And here you can also see the various types of courses available. And once you go through these courses, you would get a better understanding of what these data types are, how to use them, what are the various algorithms actually talking about, and that would make you ready for the project that we usually do on this platform, and you can do as well. So I would also take this opportunity to show you my own project that I did in the domain of astrobiology. So if you go to the project section, you see here, this is my project. I click on it. It says the transcriptomic analysis of DNA damage response in zebrafish embryos under simulated microgravity. I won't go into much detail today because I would be doing that step by step, explaining every method and step that I use to do this project in a later session. But to give you an overview, what I did was I investigated what is the effect of simulated microgravity on zebrafish and how that affects the DNA damage response, and whether the damage that is caused by microgravity and radiation comparable, similar, or different. And that's what my project is. You can definitely check out my project on the portal itself and read all about it. So if you go here, you would also get access to the data sets. Then you can read the abstract here, understand what's the project all about and why are we doing this. It's said in the introduction section. The methods, those who already are familiar with omics and bioinformatics would definitely understand the methods that I have used here. Some of them are machine learning algorithms, some of them are not that. Here's the results that has been generated from all those applications and tools. And the final part is how I interpret the results that has been generated from the pipelines and make a conclusion about what I understand from the data that is generated. So this is all about the projects that you can do here at Pine Biotech's server platform. And as you might have understood by now, that it is pretty much important to do that, to get your hands dirty, to start working on your own project, right? Because that gives you an upper hand, that gives you an edge, and that helps you to get selected in all those opportunities. So here I would like to give you a small overview of the program that we are going to launch. It would be a 1 month program and it's divided into 4 parts, you can say. So first, there would be interactive sessions. They would be usually 1 hour live sessions with Q&A. They would be held on Zoom, just like today, and there would be recorded sessions available for those who register and book their seats as well. And after you have done that, this is for the first time that I am letting the cat out of the bag, that there would be a guest special lecture by a guest faculty, and that person is an expert in this domain. He or she has been working in the domain of space omics for quite a long time, and it's always a good idea to learn from the best in the business. So stay tuned, register yourself to get to know who that special guest lecturer would be, and I'm looking forward to this session pretty much. After you have done that, after you have gone through other courses, after you have attended the lectures, you would get an opportunity to start working on your own project. Definitely you would get a mentor support, and that would definitely be an amazing thing which I'm looking forward to, because working on a project, doing some hands-on is the best thing that you can do for enhancing your education, or that's what I call as proper learning. And finally, when you have done that, you have completed it, congratulations, you have gone through this hustle and finally earned your certificate. You would get certified by Pine Biotech, and that would be an amazing feather to your hat added. So finally, I would like to give you a small overview of the 1 month program and what we have in store for you. The first day would be an introduction to astrobiology, talking about what this term means, talking about origin of life on Earth, the various extremophiles. We would also touch upon the debate of RNA world versus DNA world, the tree of life, and it would be on 19th of October. On 25th of October, we would talk much more focused on origin of life and evolution. We would talk about the great endosymbiosis theory. We would show you how you can work with data sets, where you can find those data sets, and we would also talk to you about some project examples of reconstructing ancient RNA or ancient protein using machine learning. There has been a lot of work which has been done on a very popular protein which is called Rubisco, to recreate it, reconstruct it, understand how it evolved, and that gives us a better picture of origin of life. Then on 1st of November, we would talk specifically about space omics and astronaut health, again taking the reference of the NASA twin study, digging in deeper, understanding the various environmental factors, understanding the various results of this study, and I will also show you how you can retrieve the data and get ready for your project. And on the 8th of November, there will be one session which would talk more about the various omics types which are required for space omics and astronaut health, like genomics, proteomics, transcriptomics, metagenomics in general, so that you have a better picture of what we are dealing with. And finally, on the 15th of November, we would focus on a case study of a few projects, including mine. So I would give you an overview, step-by-step analysis of my projects, the methods that I used, and the results, and how to interpret them. So that's all that I had for you today. I hope that you got some insights into what we are dealing with and why we are so much hyped up about this particular field of space science. And I hope that you liked the fact that we are going to provide you an opportunity to do your own project so that you stand out from the crowd. So I think that's all that I had for you today, but I will transfer the stage to Sonalika, who would give you an overview and a brief understanding of how you can register and be a part of this program and the other details. And also, if you have any questions regarding space omics or astro